Okay, this is key assessment 6B, question 5. And already before I've even read a word of the question, I can tell this is about cell division because I can see a root tip squash there. And just a quick glance across and I've got a graph of DNA in arbitrary units. So I'm already thinking cell division. It's all there at the back of my mind. So, colchicine is an alkaline compound derived from the corm and other parts of the autumn crocus. Do not eat these, they're really poisonous. Colchicine induces the disassembly. No, disassemble. Sorry, number 5 alive moment there. And thus stops the mitotic process, so stopping mitosis so that chromosome, morphology, shape and number can be studied. The photograph shows a preparation of garlic root tip. So we've got a root tip squash using colchicine, showing the different stages. No shocker there then. So here we go, that's anaphase, really recognisable, really easy to know. Squash spider, that's for metaphase 1. And then I've got 2 clumps, so that's 2 telo. So remember, anaphase is moving apart, 2 clumps, and these are in the middle. Name the stages shown in A and C. So we're only looking at that and that. So don't, just put them all down just because you've super duperly identified them all. So A is going to be telophase. And stage C is metaphase in the middle. That's something that should be in your head and you've done it in the practical. Explain. So we're going to use a because word. Why the disassembly of microtubules, short circuit, great film, would stop mitosis. Why would it stop mitosis? So what do we know? Microtubules. So microtubules form the spindle. So spindle cannot move chromosomes, and that's why it stops mitosis. Obviously, if they're sort of, attached to the spindle in the middle, or they're being pulled apart when you apply the colchicine, they're going to be stuck there. Oh, there's another little anaphase nucleus there. So interesting, root tip squashes. So that's what stops it. Explain. So explain words, so because. So there's my explain words, so there. Oh, and we're explaining. So, and explaining pretty often turns out to be KA2, I'm thinking. Explain. Again, explain the purpose of the process occurring during the stage labelled B. So it's saying, well, why do we bother with the anaphase? And pretty much I'm thinking that's something that you should have in your head. Possibly because you're explaining it. It's an KA2, but it could be KA1 because KA1 includes explanation. So, the identical sister chromatids are being pulled to opposite poles. So, it's my explain word, so the cells formed will be genetically identical. So that says what's occurring, why would we bother? Okay. quantity of DNA in the nucleus of an animal cell was monitored for a period of 4 hours, as shown below. So we've got time along the bottom, 4 hours, DNA, trundling along, doubling, staying the same, halving. So half, double, same. And probably your teacher went through this graph. It's a pretty standard graph. So what's happening here? This is the DNA replication. When does that occur? Interphase. Then you've got pro, meta, ana, tela, and cytokinesis at the end. And of course, if you've done any past questions, you might realise that that could be meiosis 1 or mitosis. You can't really tell the difference because you get DNA replication before meiosis as well as mitosis. So name the stage, which is represented by the time period labelled X. So what's going on there? The stage is interphase. So again, that's AO1, you should just know that. Using evidence, give a reason. How do I know it's interphase? Because, and I need to use evidence from the graph. The quantity of DNA doubles from 6, presumably to 12, it's doubles, and DNA replication happens in interphase. That's why I chose interphase. So, interpreting a graph, AO2. What additional evidence would be needed to conclude if the cell is undergoing mitosis or meiosis? And give a reason that you've said that. So, if we were going to distinguish between meiosis and mitosis, obviously mitosis, if you carried it on, mitosis, that would happen again, wouldn't it? You'd get that same pattern again if it was mitosis. And if it was meiosis, it would go down to 3. That's what we would expect. So meiosis, 2 cell divisions, and it quarters the DNA from 12 to 3. So you would have to, what additional evidence, what would we need to do? We would have to observe for a longer period of time. So you'd have to let it go through all the way through the interphase and growth and what have you until then. Then if the DNA doubles, it would be mitosis, and if it halves, it would be meiosis, because meiosis has 2 divisions. So there we are, explain. I've got my because word in there. Always aim for that. Oh lordy lordy, I recognise this. This is good old Messelson and Stahl's experiment. Experiments conducted in the 1950s proved that DNA replication is semi-conservative. So already I'm thinking now, okay, so I've got my original DNA, I've got my new nucleotides, so I get splitting those into 2, adding new nucleotides in to give that hybrid DNA, and splitting those 2, and adding my new nucleotides in. So that would be generation 0, 1, 2, can't bother going any further. What have we got? 0, 1, 2, we've got third one. My life's too short. The experimental procedure involved growing bacteria with the heavy nitrogen, so this is the N15 colour, until the entire DNA contained N15. They were grown for several generations in culture medium with the N14 isotopes, so all my black ones on my diagrams are N14. At each stage they're extracted and separated and then spun down, and you get the lovely results, and there's your DNA. So this is all heavy, N15. This is hybrid, which is the ones that are composed of both, so that's that one. And then we've got these two are completely light, they're there, and then we've got the hybrids again. And then here you can just see very faintly that sort of hybrid line across there. So we've got less of that, and we've got more, the same amount of that, and we've got more light. So that's what's going on in the experiment. So, on to the questions. Name the part of the DNA molecule that's got the nitrogen in it. AO1, very straightforward, nitrogenous base. I'm going to put that word organic in there as well. That's to cover all bases. Ha! Oh, never mind.
Okay.
Oh, that's a really pathetic laugh. Anyway, it was a pathetic joke. So, state, what is meant by semi-conservative DNA replication? Explain how the results shown above support this theory. So we're just going to have to turn 5 marks. We're going to have to say 5 different things about that experiment. So, semi-conservative, let's start with the easy stuff. Then I know I've got a mark. Semi-conservative replication is where each strand of the DNA molecule, you've got to be careful using the strand word, strand of the DNA molecule acts as a template to attach new nucleotides. So that's what semi-conservative means. How are we forming evidence? In generation 0, so start at the beginning, in generation 0, all strands are heavy. The heavy strands form a template to attach new light nucleotides to. Forming hybrid intermediate. Right, what are they telling us that this is? I'm not saying. Should we say mass? Intermediate mass. DNA molecules. In generation 1. I don't think they leave you enough space to write the piles of information you need to do. We now need to deal, we've got 2 more generations to go yet, so I'm thinking marking wise, it's probably one for defining semi-conservative, one for generation 0, 1, 2, 3. So, generation 2 both light and intermediate molecules. In equal numbers or equal, what do I use the word amount? I'm going to use it. In equal amounts as the heavy strands template gives intermediate, and the light strands form templates leading to light DNA molecules at the top. Crikey! So, last generation, generation 3. We're running out of space on there as well. I'm just going to move that up a bit. In generation 3, there are more light molecules as there are more light template strands. Okay, I'm losing the will to live now. That is it. That's what I know about semi-conservative replication. You'll just have to put up with it. Why is